Good morning, colleagues. My name is Willard Dere from the University of Utah Health Sciences Center, and I have the great pleasure of moderating today's panel discussion entitled Clinical Trials, Legal and Ethical Issues in the Age of COVID-19. 11 million individuals affected in the United States, those who we've diagnosed at least, 250,000 deaths. This pandemic has had a staggering toll in human life, the economic impact on families and our broader society, and the potential long-term effects of infections in individuals, the so-called long haulers. This period over the past 9 or 10 months has also brought unprecedented public-private partnerships with national initiatives such as Operation Warp Speed, which has helped assist bringing vaccines, now first one being available, potentially available under emergency use authorization. That Operation Warp Speed appears now to be bearing fruit. This particular pandemic has raised a number of potential issues in the clinical trials arena. We have, as I said, an unprecedented number of public-private partnerships, national and academic centers working together with industry. We've actually had a large number of clinical trials that have been implemented very, very rapidly, and in fact, unauthorized or unregistered medications being brought to patients without the usual evidence base that we usually use in the field of medicine. We've actually had questions about patent protection in a setting of unprecedented national emergencies with the public-private partnership. What is the patent type protection for the new platforms such as messenger RNA? We've had questions about clinical trials. How do we, for example, at the University of Utah, prioritize trials? Do we do single center trials, or do we participate in multi-center trials that potentially can answer key questions, as the UK has done with the use of dexamethasone in the setting of the intensive care unit? And can vaccine trials be implemented more rapidly, even that they have done so to date? Moderna with 30,000 clinical trial participants, Pfizer with 44,000 clinical trial participants has been implemented very, very rapidly, but could we even move faster? And actually, how do we ensure that the benefits and risks of available vaccines can be made available and are safe in the underserved population? One of the reasons why this is such a great pleasure to moderate is because of our panel of experts, who I will introduce in the order of their presentations and move to the first speakers. But the panelists are Dr. Mike Dean, Professor of Pediatrics and Associate Dean for Clinical Research at the University of Utah. He will be followed by Dr. Erin Rothwell, who's Associate Vice President for Research Integrity in the Office of Research Integrity and Compliance, and a professor in the Department of Obstetrics and Gynecology at the University of Utah School of Medicine. Ms. Seema Shah, who is Associate Professor, the Founders Board Professor of Medical Ethics in the Department of Pediatrics at Northwestern University's Feinberg School of Medicine, and Associate Director of Research Ethics at the Lurie Children's Hospital. And finally, Ms. Ana Santos Rutschman, who's Assistant Professor at the St. Louis University. So I will now leave it to the panelists and start off with you, Dr. Mike Dean.
Thank you, Will. My goal in the next probably 10 minutes is to just lay out some vocabulary and give the landscape about clinical trials so that the legal and ethical issues can be discussed by subsequent speakers. What's the definition of clinical research? Some people think that clinical research is always trials with drugs or devices. I include anything that involves patients or humans as clinical research, and some of these types of studies are not interventional. Some of them are studies that can be done without even encountering a patient. You can do work with medical record review, you can do cohort studies and so forth. I think the relevant categories of trials that we might talk about today are notably focused on COVID-19, but I will raise a question about whether other clinical research is being crowded out and whether that poses any kind of an ethical question. Categories of drugs in COVID-19 are confusing, and you will read papers or read announcements that such and such a drug has been definitively proven to not work in COVID without definition of whether people are talking about outpatient interventional trials, inpatient interventional trials, and if inpatient, are they talking about patients that are on the ward with relatively moderate disease, or are they talking about patients that are in the intensive care unit on ECMO machines, which are extracorporeal membrane oxygenation machines? These are all very, very different levels of severity, and there is good evidence that the severity of the disease might dictate what types of therapies are going to be beneficial. Cohort follow-up studies are an interesting question because normally we would consent patients to do follow-up, but there is an exemption from consent requirements, and Dr. Rothwell may speak about this later, based on public health mandate. And so there is a little used exception in the law that says that a public health agency at a federal level can decide that research, including follow-up, can be done without consent if there is an imperative from a societal standpoint. I've never heard of this actually happening, but it is in the CFR. The types of interventions that you'll read about or hear about in terms of COVID include antiviral medications. Remdesivir is the most well-known of these, and remdesivir has been approved for use based on a trial that was stopped prematurely, in the opinion of many people, because it reduced the duration of hospitalization by approximately 30%. Interestingly, this morning in the newspaper, there was a description in New York of the length of stay of patients with COVID-19 already dropping from the spring until now by about 50% just because of better care. So I think that whether remdesivir works or not will be an interesting question for us to see over the next few months. Convalescent serum is a therapy that has been used for over 100 years. Based on the idea that if somebody survived, they must have some kind of antibodies that help them survive. And so you harvest their serum and you give it to others. Today, we're much more equipped to identify antibodies and have an idea that they really are relevant to the virus. And there are numerous convalescent serum trials ongoing. Dr. Dere mentioned dexamethasone as an anti-inflammatory, and dexamethasone, like other steroids, is a drug that gets applied in every disease and works in some of them and doesn't work in others. I think you'll recognize potentially that President Trump received all three of the drug categories that I just described, but I would have to emphasize that none of these three drug categories have been demonstrated definitively to work. There are antithrombotic drugs that are used because this virus causes in some patients enormous amounts of clotting and thrombosis with complications such as strokes. And if that happens, they need to be put on anticoagulants. And then, of course, vaccines. And I'm not going to speak about vaccines because a future speaker is going to go into detail about this. We have worried in Utah about competition between different vaccines. If you think about a population in Utah receiving different vaccines in different trials, you are affecting the background infection rate. And you're doing it in a way that could be very confusing to the results. But I think given the results of the 2 US vaccines in the last 3 weeks, this may become a moot question. That is, these vaccines may be put into production and used. I would be amiss if I didn't mention the famous hydroxychloroquine debacle, in my opinion. Hydroxychloroquine is a anti-malarial drug. It's used in a variety of rheumatic diseases. And there have been observational studies suggesting a lower rate of infection with COVID in patients that were receiving this. It's an interesting drug because had President Trump not mentioned it or endorsed it or placed wild claims behind it, we might be investigating this drug in a more scientific manner. But I think that it has really become very politicized whether this drug works or doesn't work. The evidence is pretty good that on the inpatient side, it does not work. And earlier this month, there was a release from Intermountain Healthcare and the University of Utah of one of our trials of hydroxychloroquine that indicated it definitively does not work. But the news announcements didn't indicate that this was an inpatient trial. And it also didn't really get into the fact that there were only 85 patients that were in the trial. And it's far from definitive that it doesn't work. It did not work in this particular trial, but there weren't enough patients in this trial to really prove it. So you can ponder whether our attitude about the drug would be different if it had not been politicized. Finally, what's the impact of COVID on other clinical research? At the University of Utah, we placed very stringent restrictions on clinical research unless it was cancer-related or life-threatening to not have clinical research. So there are certain trials where patients are dependent on receiving investigational drugs or devices. The reason for this was because we had a lack of personal protective equipment. We did not have enough masks. We also didn't understand the virus. We didn't have adequate testing. And for that reason, we shut down clinical research unless it was basically oncology for chemotherapy agents. We know a lot more now, and we have adequate supplies of PPE, which is the personal protective equipment at the university, to be able to protect ourselves and subjects from exposing themselves to risk. A question that has come up is, does clinical research pose a risk to participants of encountering or catching COVID that should be disclosed in consent forms? Several program officers from the National Institutes of Health, or the NIH, have asked this question of some of our investigators and have said, does your consent document suggest or include the risk of catching COVID? And our response back, after a lot of thought in the dean's office with respect to the research environment, is that if we thought that there was a significant risk of COVID from participating in non-essential clinical research, we wouldn't allow it. We believe that in the context of PPE and proper social distancing and all of the things that we're doing in our research areas, that the risk of contracting or transmitting COVID is essentially nil. We've had no known transmission of COVID from any clinical research visits. And in comparison to the everyday community risk that patients have, we don't see an incremental risk in participating in clinical research. And my final thought to leave with you is that if clinical research participation in general confers any benefits to subjects, think about that. You're in a trial, you could have the drug or the active agent, or you could have a placebo or usual care. But there is a perception in the research community that is backed up by significant data that mere participation in a clinical research trial confers benefit on the patients that are getting the placebo or the new drug because of the additional attention that is paid to those subjects. So if clinical research confers any benefit to subjects, is restricting clinical research ethical? Unless there's an incredible risk that we can point to the research as being a risk. And since we believe that there's no incremental risk from participating in research, we believe that there are some ethical questions about whether shutting down would make sense. And this has come up in the last 2 weeks, obviously, because of our surge. To date, we have not shut down clinical research. I think if our facilities are overrun and there's no place to do the clinical research, this may be another angle that's going to complicate things. With that, I'm going to turn this over to Dr. Erin Rothwell for her comments.
I have a slightly different angle here. I'm going to talk really quickly a little bit about just an overview, some of the key legislative mandates that set the foundations for clinical research, talk about some of the issues that have arisen during COVID, really focusing on 3 specific contexts that I've had direct experience with, and then of course open up for questions and suggestions. So I know this is a lot of words, but I'm just going to give you a different version of this. But you know, in 1938, the Food, Drug, and Cosmetic Act was one of the first key legislative mandates in the US that required human trials to have safety data before marketing to the public. This was then quickly followed, I mean, not quickly, but this was followed by the Nuremberg Code after the Nazi war crimes. And then 2 days later, the Declaration of Helsinki provided the first international set of ethical principles for research with humans. And then the US Surgeon General that same year gave the recommendation that there is a need for independent review of research. So in 1974, the National Research Act that required that any research funded by the US government must undergo an IRB review process, as well as a requirement for funding. And it also laid the foundation for the National Commission for Protections of Human Subjects, which produced the Belmont Report, which is going to be a key, I think, piece of legislation or piece of documented evidence that is going to really articulate the foundation for the rest of this discussion. And we all know that the ethical principles that came out of the Belmont Report, respect for persons, beneficence, and justice, became the basis for the federal policy for protection of human subjects, otherwise known as 45 CFR 46. Then this became known as the Common Rule that was codified in 1991. And then in 2017, the updated Common Rule came out, and really, I think one of the biggest changes was focusing on consent and what a reasonable person would want to know. And in my opinion, that just reiterates that the current consent process prior, where we have these lengthy, dense documents, really did not promote or were not effective in promoting informed decision-making for clinical research. So when we think about what makes clinical research ethical, you're going to see that, you know, a lot of scholars have looked into this. You know, right here, the Belmont Report lays out 3 things that state that these need to be present for the clinical research to be ethical, to justify the use of humans in this type of experimentation. But really the goal of clinical research is to develop generalizable knowledge for future patients. And that knowledge is added to the evidence base for us to, you know, develop new treatments, new protocols for managing specific diseases as well as care. Emmanuel came out with another list of statements in 2000 about what must be met for clinical research to be ethical. And again, it's very similar to the 3 components of the Belmont Report on the previous slide, but you'll notice that, of course, informed consent is being raised, independent review, and then, of course, the potential to recruit multi-regional clinical trials for participation. And then the most recent one that has now revised and come forth and said, well, what makes clinical research ethical? And this came out in 2016. There's 11 key essential elements. And this has been really pushed hard out of the multi-regional clinical trials center led by Barbara Beer at Harvard. And I think this is starting to capture the complexity about how can we ensure that we are respecting persons, you know, beneficence, and ensuring justice in terms of access and participation. And you'll notice that community engagement and return of research results is somewhat contemporary compared to what was discussed before. So why are these frameworks continuing to emerge? We can go down a list. You know, obviously, research misconduct is definitely a cause of that, but also the globalization of clinical trials, increased awareness about clinical trials. You know, individuals have access to more information, you know, much more readily than they did 40 years ago. But really, you know, I think one key point too for why these frameworks are continuing to emerge is not a lot of people are aware, but the Declaration of Helsinki was updated in 2013 that medical researchers, that medical research conducted by individuals should have appropriate training and qualifications in clinical research, which when I was discussing this presentation with some colleagues, they found this to be quite alarming in terms of, well, couldn't anybody conducting research, wouldn't they already have all the training and competency to be engaged with humans and experimentation? And not necessarily. And I don't know why this is not moving. So, this came out in 2014 out of Center Watch. And I look at this graph here, when we move from compliance to competency, and I keep thinking about Mike Dean's shop in Department of Pediatrics, the Data Coordinating Center, and I'm thinking one clinical research professional cannot entail all of these requirements for competency, that, it really is team-based. And I see a lot of these coordinating centers fulfilling a lot of these requirements in order for research to be carried out not only effectively, efficiently, but ethically. So that could be some of the reason for this continued examination about questions about what makes clinical research ethical. Before we jump into some issues specific to COVID, one of the things that I have found, clinical trials are critical. They are critical to the evidence base. I do like Dr. Dean's comments before about, I think clinical trials, clinical research is any human involved in research. But what we found is a lot of the efficiency of clinical trials have not reached expectations. They have not contributed meaningful to the evidence base. And that means participants are exposed to risks and burdens without fully knowing that their participation is not contributing to medical progress. And I'm not saying this is all clinical trials research, but in general, you see clinicaltrials.gov coming out and asking for investigators to post their trials, requiring it with fines because of poor communication. And that means, a lot of trials are being conducted that are non-informative or not shared, and they're taking valuable resources for other trials that need to be conducted to really add to the evidence base. And to specifically kind of target US medical centers, the biggest critiques are that sometimes the clinical trials are not well designed, they're not well thought out, and the results are not reported. And when you think about the insufficient use of trial data to inform research decisions, that's also the initiation of new trials, right? So we have a lot of competing trials with no clear guidance on how to prioritize within our own academic centers. And this was quite, I think came out during the COVID pandemic. So in the age of COVID, which I'm being hopeful that it's coming to an end, I think we're still learning. We're still trying to figure out what, how are we conducting research, and how can we learn from what we've been doing wrong in the past to this, I would say, kind of real world laboratory. And a lot of the problems that we're facing now with the implementation of clinical trials were present before, but there's also these new expectations. The public is demanding some type of treatment for COVID, right? And you've heard a couple comments before that we're trying new treatments, we're learning a lot more, we're advancing, and it's proving to be, I think, a positive light on clinical trials. But the way that we're conducting clinical trials during COVID is not the same that how we were conducting them before, with long study startup times, and then of course, lack of reporting of results for others to learn from. And so, I think in general, the issues that have been raised with COVID have been there previously, but it's also forced investigators to work outside the box and to push things. I think, I already mentioned Dr. Dean previously. I'm only going to pick on him because he was the person before talking, but his data coordinating center got a trial up in 2 weeks, an outpatient COVID trial, right? Before that would have taken 6 to 12 months. And so, how do we replicate what was successful during COVID for the future? However, I think there's a wide variability in the types of trials that are going on in COVID. not only outpatient, inpatient, but we're also now facing vaccines. I'm sitting on the board right now for how do we distribute the vaccinations to healthcare workers? How are we prioritizing who gets it first? And that tends to be a little easy, but the second phase, the third phase, who gets it next and next, it's quite complex. And so, these issues, I think I'm hoping we continue to learn from so that when we're faced with another pandemic, or if we're truly going to be, I think, changing the clinical trials enterprise based on the lessons learned from COVID, we can take those key recommendations. But I'm going to talk about three things that I have noticed that have come out during COVID-19. One of the big things is that the protocol development is not transparent, right? I can't tell you how many protocols I've read on this therapeutic panel where, and in general, or with my office, that people just add this blanket statement. This study will be performed in accordance with ethical principles in the Declaration of Helsinki. And then that's all they say. And then they have this huge long section on informed consent. And I hate to say it, but informed consent is not working. It's not effective in promoting informed decision-making. So if we're relying on informed consent to be their ethical pillar for what makes clinical research ethical, we're really missing the boat here. And so when I'm reading a protocol, I don't understand how individuals or these investigators came to these decisions of using a placebo when the same trial, very similar in an outpatient setting, is not using a placebo. They're using a different type of interventional drug. And it's just not clearly laid out. And I think one of the other issues about protocol transparency that has come out is, how are we making sure that we're ensuring equitable access for non-English speaking participants? And do we have a plan to ensure that the participants we enroll are representative of the population we hope to have these results return to and to implement for their care? And right now, when you look at protocol developments, a lot of this poor transparency, I think, is leading institutions to come up with their own mechanisms for prioritizing which clinical trials should move forward first. And I have an specific example of that here shortly. I do want to touch base again on informed consent, because this is because of my research agenda, but I'm leading the NCAT's informed consent group. And one of the biggest things that have come out is this new awareness among investigators that, wow, informed consent really isn't promoting informed decision-making, right? It's more of a mechanism to try to capture a signature. And we are having difficulties capturing a signature because of potential contamination. And, people are using RedCap, people are using DocuSign, they're having people take pictures of their signature on their phone and emailing it to research teams. And the clinical team is the one engaging with the consent process. It's not the research team. So we're starting to see this awareness among two different stakeholders. Wow, we really need to rethink how we promote informed decision-making, because if this is what we're using to ensure that this person is being consented in an ethically appropriate manner and understands truly what the research is about and what they're agreeing to participate, then we might be in trouble here, and we need to rethink that. And so my final topic here, and I'm going to give you a little bit of a case study, is, knowing that potentially maybe informed consent is not highly effective for promoting decision-making, knowing that we're really lacking some data on how protocols were developed, how these key decisions in the design and the recruitment of participants were chosen, that leaves academic medical centers to prioritize COVID trials. And so when I share these next couple slides, we at Utah, at first, before the surge, we had a very limited number of potential patients who were sick in the ICU at the University of Utah. So we had a high number of trials that were trying to recruit these participants. So we were kind of stuck in this position of, if this participant enrolls this trial, they're excluded from this other trial. And we don't know which intervention works, right? We have hydroxychloroquine, we have this other interventional drug. and this urgency to try something. And I would also say, not only was there this urgency to try something, but if we did not enroll every single patient into a clinical trial, then that was a missed opportunity to help find the best intervention for future patients. So the goal of this committee, which I'm part of, was not to rank trials, but it was to categorize potential COVID trials into 3 different buckets: top tier, middle tier, low tier. And so how do you come up with a criteria? Knowing that background that I just gave you about what makes research ethical, how do you come up with a criteria to categorize which trials should be offered first to a limited number of patients? And then second, how do you offer multiple trials? How do you make a decision about that involves the patient or the study team or both? So here are the 10 questions that this therapeutic panel, and of course I helped contribute to this too, and it is a work in progress, but it's a starting point. And I have talked to other institutions. We did this huge CTSA survey, and other institutions are doing very similar things to help prioritize COVID trials. So University of Utah is not unique in this. And one of the first things that we've asked is this trial sponsored, right? And so if you can just start thinking, what do you think is the relevance of asking that question? So is a trial less, I would say, important if it doesn't have NIH funding or external funding, and it's just purely investigative driven? What is that? How do we infer that? And we did rank external funding higher than no funding for a trial. And it wasn't so much the importance of the study, but it was also the resources to be able to carry out the research. Because again, we're already overworked with clinical care, but we also need to make sure we have the resources to support the research. The other—
1 minute, Erin. 1 minute,
yeah.
please.
Sorry, okay, 5 minutes. Then it was prospective and randomized. The other one was multi-center. If it was multi-center, it was ranked higher than if it was only conducted at our own institution. Of course, we have all this other data, which I think is clear that, if there is a yes to this component, then of course that tends to have more weight, more validity. And then finally, was there a control group? Was there an active control group? Was it statistically powered? Would we get enough participants? Would it really recruit enough in order to make a difference? And then this is a unique one here. Was our own institution engaged, right? And if yes, they got like a tiny point towards that. And so that just raises another level of some considerations around the ethics of, prioritizing COVID trials. And again, these were not to rank, and they were very loosely used. So most of the trials were up in the top tier. It was up to the study team and clinical team to decide which trials were actually offered to patients. So in summary, I'd like to say that the pandemic has provided a real-time laboratory. It's very characterized by rapid movement, some uncertainty in treatment, and there's a significant urge to keep trying stuff. But as this pandemic persists, we have so many opportunities to learn and evolve our clinical trials enterprise. I'm energized to rethink how we do consent. I'm energized to rethink how we do study startup. And it's time to let go of this 1980s style approach that I've heard a lot. Like, I think that's the number one comment I've heard about consent, about, it really opened our eyes that we were cemented into decades long approaches to clinical trials, activation, as well as consent. Thank you.
Okay, our next speaker is Professor Shah.
So good morning, everyone. It's delightful to be here. And I think what's really great is I think this symposium is incredibly timely. So as you'll hear from myself and the next speaker, today is the day that 2 vaccine researchers have announced their intentions to submit for an emergency use authorization. And many of us, I think, are hoping that this is the light at the end of the tunnel for this pandemic, although we still have a long way to go. So what I'm going to be talking about is accelerated vaccine development in the COVID-19 pandemic and whether controlled human infection trials should be deployed. And I'll try to build on the ethical considerations you've just heard about. So first, to make sure that we're all on the same page, what are controlled human infection studies? And these are somewhat controversial studies that have actually been around for hundreds of years. And they're also referred to as human challenge trials or controlled human infection models. The basic idea behind these studies, no matter what name you use, is that these are studies that deliberately expose participants to the pathogen in question. And this can be done either to rapidly accelerate vaccine testing, because instead of waiting for people to become naturally infected, they're deliberately exposed, and you can compare people who got a placebo vaccine to people who got the actual experimental vaccine candidate very quickly. They can also tell you a lot about early phases of diseases where people might be asymptomatic and wouldn't otherwise present to care. And participants are followed very closely and in a highly controlled manner. They've been done for decades on typhoid, cholera, malaria, and have been done quite safely. But as this picture illustrates, they can be very burdensome. So this man was a participant in a 1970s University of Maryland cholera study. And the reason he's holding up that sign that says 26 is he needed 26 liters of intravenous electrolytes to replace the fluids he lost after becoming infected with cholera, but was, of course, fine at the end of the study. So I think it's useful to understand these studies in terms of how they fit within the traditional vaccine development pathway. And traditionally, the way that vaccines are tested is a lengthy process and a very careful one that involves first testing the vaccine in small numbers of healthy people, just to see whether it's safe and if there's some sort of immune response that it generates. The second phase is to test safety and to preliminarily look into whether it works by testing it in people who are being exposed to the actual disease in question to see whether the vaccine might start to protect them. But protection and efficacy is really tested in phase 3, where tens of thousands of people are given the vaccine or a placebo, and then researchers wait to see whether the vaccine protects against severe disease or infection. In the context of COVID-19, that's involved upwards of 40,000 participants in the individual trials. And once that happens, once that entire package of data comes together, then it's submitted for regulatory review. Now, ways that people are looking to accelerate this pathway. This has been sort of an age-old question, really. So this figure comes from the Zika virus pandemic, and the COVID-19 pandemic has put even extra pressure on this pathway. But one possibility is to really try to compress the pathway. So to either eliminate phase 3, or in the case of the COVID-19 pandemic, people have really been focused on moving things not in parallel, not sequentially as you see here, but in parallel. And overlapping the phases of trials, not waiting to analyze all of the data before moving forward to the next phase of testing. The last strategy that's received a considerable amount of media attention is using human challenge trials. So the idea would be to test the vaccine up in the same way up through phase 2, but then for the phase 3 efficacy part, human challenge trials would be used instead. And instead of using 40,000 people and waiting for people to become infected naturally, human challenge trials would involve 15 to 150 people who are deliberately exposed in the lab. This, of course, would require additional human safety studies, but the idea is that maybe this could accelerate vaccine development. And the final way that I think is really important to note that is being used to accelerate vaccine development is an emergency use authorization. I think the next speaker will touch on this as well, but an emergency use authorization can streamline the submission process for regulatory review. One function is that it can eliminate paperwork. Another is that it allows the FDA to use a lower threshold for authorizing the availability of a vaccine. So they just have to say that it may be effective, and then they can authorize it for use in certain populations. In a paper that I wrote with former colleagues from the NIH that was recently published in Vaccine, we analyzed carefully the different ethical trade-offs involved in expediting clinical trials in different ways. And I won't say much about this here because I really want to focus our attention on a specific proposal. But in terms of speed, the numbers of people who are involved, the risks to those participants, the cost, feasibility, and the risks of distrust in the public from using these different approaches, we concluded that expediting clinical trials was really the best way to go. And the primary reason is because it gives you the most amount of data that can help inform whether it's worth giving the vaccine to the world's population, all of the people who need it right now. But what I want to do now is focus on controlled human infection studies in particular and drill down into the ethical trade-offs that they pose. So before COVID-19 hit, there was this long history of doing controlled human infection studies and fairly limited ethical analysis, but this sort of tentative ethical consensus. People thought that these were not ethically distinct research, that in all research, as Dr. Rothwell mentioned, there's a balance between risks to individuals and the production of generalizable knowledge that will help future patients. And we ask people to take on risks for the benefit of others in the future. Controlled human infection studies, although they may feel different or maybe even cause people to have the ick factor, they really aren't ethically different in this way. There was a sense that some controlled human infection studies are too risky to permit, however, and the general criteria people would use to determine when that limit was met was whether there was a curative treatment that was available or if the disease was self-limiting and likely to resolve. However, it's interesting to note that influenza controlled human infection studies are done, and they don't really meet these two categories. Unlike COVID-19, however, influenza is a disease that we know a lot about. Finally, people agreed that there were several ethical considerations to apply, and that those ethical considerations are mostly the same ones that most research requires. And then the COVID-19 pandemic hit. And in this pandemic, because the world really was affected in a way that we haven't seen for many years, or really in the living memory of anyone who's alive today, pretty much, there was this renewed attention and interest in conducting controlled human infection studies. And two articles quickly came out in early March to say that these studies should be used to replace phase 3 trials. This has led to some movement. So there are now public plans for controlled human infection studies. The NIH has started developing strains for SARS-CoV-2 challenge trials, SARS-CoV-2 being the virus that causes COVID-19. But the idea is that these are trials that are a backup plan. They're going to be used in case they are needed. And Tony Fauci has said these are not even a plan B. These are a plan C or a plan D if the vaccine trials just don't work out. However, the UK government and Oxford researchers have planned to start testing a challenge model in January to complement phase 3 trials. And the idea is that they are still proceeding with their phase 3 trials, but they're also going to develop a challenge model at the same time. So in the meantime, after the pandemic hit, I had been engaged with a group of controlled human infection study researchers and ethicists and legal scholars really interested in developing a more rigorous and comprehensive ethical framework for controlled human infection studies. We weren't satisfied with the tentative ethical consensus I presented earlier. And this is a picture of us pre-pandemic in Switzerland, where we met to develop this comprehensive ethical framework. So we rapidly repurposed it for the controlled human for the COVID-19 pandemic, and published an article in Science, and also collaborated with the World Health Organization to develop a key criteria document. We also recently came out with a special issue in the journal Bioethics, where we sort of drilled down into many of the specific big questions that arise with controlled human infection studies. But I'll give you just a quick flavor of our conclusions. We landed on an ethical framework for controlled human infection studies that has 7 components. So we thought it was important to first make sure these studies are justified in that they have sufficient social value and a reasonable risk-benefit profile. And then other considerations are also important, like context-specific community engagement, fair participant selection, suitable site selection, robust informed consent, and proportionate payment. We didn't if you were interested in hearing more about payment, I would recommend work that's been done by Holly Lynch with this report that's available at SSRN. But for this talk, I'll just quickly focus on social value and the risk-benefit profile for controlled human infection studies to address COVID-19. So social value is the idea that it should be determined in advance that the magnitude, distribution, and likelihood of health benefits from research for populations is sufficient to justify the research. And usually this is sort of an ad hoc determination that institutional review boards make. But we think that higher risk and potential controversy from these SARS-CoV-2 controlled human infection studies requires more rigor. And in particular, it's important that there's an ability to delineate a path from doing the controlled human infection studies to obtaining important health benefits. And there are several different paths to social value for vaccine testing. I think the first one out of the gate was a little bit misleading about the best use of controlled human infection studies. So this path, which was proposed by Neer et al., Peter Smith, and Mark Lipsitch, was to replace that phase 3 trial. Although this is an interesting idea and it's sort of easy to understand and digest, it runs into a couple of key problems. The first is that regulators may not accept these data because we're only talking about 15 to 150 people who are young and healthy to make sure that these studies minimize risks. So generalizing from that population to the entire US population is really problematic. And it's not clear that knowing that a vaccine works in that population will tell you enough about whether it can work in the broader population, including older adults. The second issue is timing. So we've heard really exciting news about vaccine development, but it's just proceeded so quickly that the fact that a challenge trial takes quite a bit of time to launch meant that challenge trials were running behind from the very beginning and never did catch up. So it seems like now we're likely to have vaccines approved under an emergency use authorization relatively quickly, and a challenge trial, even just to come up with a dose that is appropriate for testing in the model with vaccines, that part still hasn't happened. So challenge studies really can't serve that goal in this pandemic. However, they could still be useful both in terms of identifying correlates of protection or being able to tell with biological markers when people are protected against COVID-19, which could be useful for future vaccine research. And relatedly, it's really important to prioritize vaccine candidates going forward. Although we do have these two very promising candidates with excellent efficacy data so far, there are over 200 in the pipeline. And we'll need at least 4 to get to a point where vaccine prices come down. Although there are, of course, as you may hear in a moment, some agreements between the government, US government, and vaccine manufacturers. But additionally, the first vaccines to be licensed still could be imperfect in various ways. One big problem with the current vaccines is the requirements for cold chain storage, which may make it difficult to keep those vaccines refrigerated at low temperatures and distributed around the world. So in order to quickly prioritize amongst all of these other vaccines, challenge trials could be very useful in getting us to more vaccines to satisfy global demand. The second issue is that once social value is ascertained, the risk-benefit profile has to be reasonable. And this means that risks and benefits to participants should be systematically identified, evaluated, minimized, and below an upper limit. Now, there are some ways in which that can be done easily. We could enroll young, healthy adult participants at the lowest risk, monitor them closely, and we wouldn't even necessarily have to nail down that there is a rescue treatment or curative treatment if enough is known about minimizing risks in this population. However, there's still major uncertainty about COVID-19. We've only been living with this disease for a year, so we still don't understand a lot about it. And there are long-term complications that are not fully understood and that seem to occur even with mild infection. You may have heard about these COVID-19 long haulers who have months and months of symptoms that they just can't shake. And there also are no treatments currently proven to reliably avoid severe complications or death. In our science paper, we tried to compare the risks of doing a SARS-CoV-2 challenge trial to other types of activities like phase 1 trials or other controlled human infection studies or other altruistic activities people engage in. And we found that they're still close. It's not clear that these SARS-CoV-2 challenge studies would be below the upper limit of what we already permit. And that's without even really factoring in this uncertainty about these complications we don't fully understand. So I think this is still a close question. Finally, I think it's really important to consider public trust in vaccines. And studies that have been done looking at public trust in vaccines have shown that only about 60% of the US population has said they will take a COVID-19 vaccine. And what's interesting is that some studies have tried to dive in to understand what the basis of this hesitancy is. We know there's already vaccine denialism and hesitancy at baseline, but the COVID-19 pandemic adds extra pressure to that. And in particular, when participants in this study were asked this question, that they worry that the rushed pace of testing for a new COVID-19 vaccine will fail to detect potential side effects or dangers, 73% of them agreed with this statement. So using challenge trials, especially to replace phase 3 trials, could really increase distrust. And it's still possible that their use down the road for particular vaccines could lead people to distrust research in general. So I think this issue of securing public trust before conducting these studies is important. So what I'd like you to take away is that there is dynamic potential social value and risk of controlled human infection studies for COVID-19, which means that it's worth investing in these, but it's still a close question on whether they're ethically justified to proceed at this moment. It will require having a robust and transparent process to determine whether controlled human infection studies are ready for launch. And even as they're launched, it's important to attend to the many other ethical considerations that they raise and the public's trust. Thank you.
Thank you, Professor Shah. And Ana Santos Rutschman is the next speaker.
Thank you very much for the invitation to be here today. I'm just going to start sharing my slides, and as I do so, I would obviously much prefer to be in Salt Lake City with you, but as it happens, I'm literally next door to what is known as Hotel Influenza, where we do challenge trials here at St. Louis University for flu vaccine candidates. So that ties into our previous presentation. And what I have to tell you today really ties into the 3 previous presentations from a slightly different angle. We are talking today about clinical trials, which in my mind, and I think most people's, are about patient populations. Secondarily, they're about drugs and vaccines that we're trying to bring to these populations, being mindful of the trade-off between the need for speed, as we see particularly in situations like a pandemic, and a host of different competing interests we need to balance with this desire for speed. I'm going to talk about this first in the context of vaccines alone, but from an instrumental perspective, and that's this idea of data infrastructure, because the collection of data, and I would submit the ways in which we then share the data we have collected, really build the infrastructure that makes it possible for these drugs and vaccines to come to market, but also for us to speed up the development of things like vaccines when a pandemic or a large-scale public health crisis takes place. So I'll start with these 2 images, and I think I'm probably not too far off the mark when I say that within a span of 100 years, these might well be the 2 most iconic vaccine races that we'll experience. So on the left side of the screen, you can see the very first volunteers for the polio clinical trials in mid-1954. And on the right side, this is Jennifer Haller, who was the first person to receive a COVID-19 shot in Seattle in March of this year. So a lot has happened between these 2 vaccine races, in which, again, the speed at which we were able to develop the vaccine candidates, but in particular, the ways in which we were so able to so quickly test these vaccine candidates on increasingly larger populations is something that from a scientific perspective is obviously an enormous accomplishment, but it's also something that we've done, as you've seen from the presentations of the previous speakers, in ways that are still very much far from perfect. So just to place this idea of building this data infrastructure on which we rely to bring vaccines to market, here's just an overview of some of the things that have changed between picture number one, so the 50s, and the day that Ms. Haller got her COVID-19 shot. I'm going to just highlight a couple of things because Dr. Rothwell in particular has spoken to this. But in 1962, the drug amendments to the FDCA introduced a requirement of a showing of efficacy, which is really the base for the clinical trial model that we have today, and the gold rule pioneered by the FDA. That is still pretty much the norm in the ways we attempt to design clinical trials. I'm going to jump to 1997 here because in my mind, the two phenomena are alike, 62 and 97, when further amendments to the FDCA required clinical trial registration. These two types of changes that we've witnessed in between these two vaccine races speak to the ways in which we produce data and record data in ways that make clinical trials, one, more comprehensive, but also more transparent, and the ways we want that data to be available, one, to researchers, but also to the public at large, which is especially important given the levels of mistrust that our panelists have already referenced that surround the field of vaccines. Now, there's other important changes, and again, most of these have been referenced already, but the other years you see there, these are other types of interventions that are more people oriented, but that I would just point out also have an impact in the ways we collect data before and through clinical trials. So from the Belmont Report, the Common Rule, and the Revised Common Rule, to the 1993 piece of legislation that technically said we should be enrolling more minority populations, racially and including also women. All these types of interventions help us look at data as the building blocks of scientific research. Except that traveling from 1954 to the COVID-19 vaccine race, we know we have not been very, very good. There's been improvements, of course, but we've not been very good at incorporating all of the changes that the medical literature and the scientific literature tells us we should be attending to. So I'll play this out in the context of the COVID-19 vaccine race in just a second, but these are some transversal problems that do not affect exclusively vaccines, but that are certainly very relevant in the context of the current vaccine race and the ones that preceded it. The first one is the underrepresentation of minority and low-income populations. Second, the underreporting of many components of data collected during clinical trials, but particularly of data indicating a possible harm associated with a health good we are testing. Transparency issues that have been hinted at, and I'll return to towards the end of the presentation. Limited sharing of clinical trial data, and I'm going to, I think, occupy most of the remaining time primarily on this idea, and then mistrust, and that's something that we are more aware than ever. The World Health Organization last year, and for the first time, classified vaccine hesitancy as one of the greatest public health threats worldwide, and we've seen levels of vaccine confidence in particular dipping in the years leading up to the COVID-19 vaccine race. So it is against this backdrop that vaccine clinical trial data is produced in what I would characterize as a patchy Apache infrastructure, because we know we're collecting data. We've overcome some of the most grievous ethical violations that were still taking place in 1954 and even after that in the context of vaccine development. But we are still not exactly very good at having a holistic, representative data infrastructure. Again, not just in the context of vaccines, but I will restrict my comments here to the way we collect data on vaccines. I'm going to use the timely example of COVID-19 vaccine trials to just illustrate the holes in our data infrastructure, and then I will spend the rest of the presentation talking about the ways in which we are perhaps not at optimal levels in terms of sharing the data we do collect, which again is important to recognize is limited. So let me take a moment to first talk about this idea of the data infrastructure as having a series of building blocks that are critical for a number of things. First one being approval or authorization of new vaccines, and then second, just for further clinical research. This is the two vaccines that vaccine candidates you hear the most about these days, the ones that are likely to be evaluated over the next couple of weeks for an emergency use authorization, and I'll say more about that in just a second. But this is the enrollment goals of Pfizer and Moderna as they started phase 3 clinical trials here in the United States. So Pfizer aiming to enroll 44,000 volunteers had the following breakdown in terms of diversity populations: 12% Hispanic, 8% Black, 5% Asian, and 0.6% Native American. This would give us a diversity rate in terms of enrollment of 27%. And Moderna, which was aiming to enroll 30,000 volunteers, at the get-go had a very similar, they didn't disclose the breakdown initially as Pfizer did, but they were aiming for a 24% diversity rate. So we have 27 and 24%. Now, as you're probably aware of, a lot of experts were worried about these numbers. The current target in clinical trial design is that the trials should reflect the differences in the population makeup in the places you're working in. So in the United States, that means that 27, 24%, that's still 20%, actually more than 20% below the diversity in our population. So we're over 20% below the diversity targets with the numbers we used in the COVID-19 vaccine race. And I don't want to suggest that this is something that could have been done much differently in the context of COVID-19. We're racing against the clock, but this is a byproduct of ingrained shortcomings in the ways we enroll patient populations for vaccine clinical trials. So this should not come as a surprise, but it's an expression of long-term problem we have yet not solved. And as a result, the data that we collect are incomplete. It might be even more incomplete if we were to abide by Dr. Fauci's prescription that actually, because of the toll that COVID-19 has had on minority populations, we should not even just be trying to mirror the makeup of the US population. We should actually be trying to double it. So if that were to be the case, diversity enrollment should be around 66%, and we're obviously way far off the market. So if you agree with this vision of clinical trials and minority enrollment, that we actually, for diseases that take a particular toll on minority populations, should change the current targets, we're still incredibly far off the mark. So this is just to put things in perspective, because we are making progress and we're collecting data. So I'm going to transition from the idea of building blocks, which is one of the things that clinical trials do give us the raw data to look at and act on it. So I'm going to look at two different ways in which we act on that data. The first one is institutional, and it involves the regulator for pharmaceutical products entering the market. And then the second one is from a follow-on research perspective. So what can we expect other researchers to do with the data that will be shared at the end of the clinical trials for COVID-19 vaccine races. And I phrased this slide in a way that's predominantly quantitative, but I want to now emphasize the qualitative angle of this question. We are generating clinical trial data against the clock. We know the data are patchy and are not reflecting, if nothing else, the diversity we would like to see in the enrolled population. But we're about to have the regulator, at least in the United States and soon enough in Europe, make a decision on whether the data that have been collected are enough to warrant an authorization, not full approval, but an authorization for these two vaccine candidates to come to market and be distributed to increasingly larger populations. So emergency use authorizations in the United States, which is the regulatory mechanism that has already been alluded to, is exactly what's on the table right now. And as mentioned before, this is a regulatory pathway that recognizes the need for speed in the review process, but also a need for speed that manifests itself by having the sponsors of these vaccines show the FDA, share with the FDA or the appropriate regulator, if we are outside the US, data that under other circumstances would fall short of the criteria that normally a sponsor of a new vaccine has to meet. So I just wanted to go through the legal architecture of EUAs very, very quickly. What they do in the US is to lower the threshold from substantial evidence that the vaccine is safe and effective. So that's the standard threshold. And when the EUA will lower it from a showing that in terms of efficacy, not safety, but efficacy, it is reasonable to believe that the vaccine may be effective. So this is the standard that we know the FDA is going to be called over the next few weeks to assess the data that have been collected by Pfizer and Moderna. And in the particular context of COVID-19, there's an environment that already infuses some mistrust into the EUA pathway. This is, again, as referenced by previous panelists, because FDA has issued a number of EUAs, which the agency does on a regular basis when there's a public health emergency, so nothing new here. But some of these EUAs, as referenced before, were based on data that were questioned by experts in the field, including current and former advisors to the FDA. And in one unfortunate case for which the FDA quickly apologized and restated the data, but there was overstatement of data in the case of convalescent plasma. In an area as fraught with mistrust as the area of vaccines, this of course is going to color the process through which we bring these vaccines to market through the EUA pathway, which I will just add to further complicate things, has really never been used before in connection with new vaccines. So COVID-19 really presented us with this new question of when do we decide that the data that has been collected, imperfect as it is enough to warrant a regulator to make a decision to bring those products to market under this lower threshold. Now, there's another angle to this, which is, remember, the data infrastructure itself has long been far from perfect, and we know from emerging studies that there have been some holes in the data generated in connection with COVID-19 vaccine trials. So this report came out 3 days ago from the Government Accountability Office. The safety data for Moderna and Pfizer's vaccine candidates has been largely uncontested, and as I'm sure you know from the news, efficacy data actually is very, very promising. That being said, the data have to be interpreted in context. So what this recent report 3 days ago showed is that these two vaccines, as part of the Warp Speed program, have followed typical clinical design, meaning they've predominantly enrolled healthy adult volunteers and excluded minorities. We've seen that before, but also pregnant women, children, and populations with certain comorbidities. Now, the data that we are about, or the regulator is about to use to authorize these vaccines, again, has several unknowns. More problematic, I think, the Gao report also found that the clinical trial protocols provided very limited details on how vaccine developers have analyzed and will analyze safety and efficacy data, again, particularly with regard to population subgroups. So again, this idea that there's a trade-off between speed and other values in the system, particularly at the efficacy level, is a question that's more pressing than ever. And in making the decision to authorize these vaccines, this is something that FDA and all regulators need to be extremely mindful of, both from a safety efficacy perspective, but also from a trust perspective in a field like vaccines. FDA's response to this, again, this was also very recent, 3 days ago. Again, FDA said they'll make available reviews of scientific data and information used in support of vaccine EUA applications, which I think is obviously a step forward, but it comes with an important limitation, which is the announcement says this publication is going to be limited to the extent appropriate and permitted by law. Now, in the interest of time, I'm going to go through these slides just a little bit faster than I would like, but I'm happy to discuss this during the Q&A or offline. FDA has a very restrictive history of interpreting data sharing provisions. In 1974, through rulemaking, FDA, and I've quoted the language used by the agency here, came to the conclusion that it could not disclose clinical trial data. So treating data, again, the building blocks of one, regulatory decisions, but also two, follow-on research as proprietary information. So FDA has for a long time held the position that it cannot disclose clinical trial data. When you actually look at FDA's organic law, the FDCA, what the letter of the law says is that FDA cannot disclose methods or processes that qualify as trade secrets. So FDA has long had a more restrictive approach to this, even after Hatch-Waxman, which has said that the public at large may actually request data used in support of a new drug application that has been granted by the FDA. So even though it seems that systemically the legislative intent is not for the interpretation to be that FDA can only keep as proprietary data that qualifies as trade secrets, that's not been how the FDA has interpreted things. From 2007 onwards, and this was a direct response to the Vioxx scandal, FDA has been mandated to share the reporting of summary results within 12 months of trial completion. So companies seeking regulatory approval have to submit these results, which again, it's a step forward, but in terms of transparency and enabling follow-on research, still limited results. And we know that more can be done because across the pond that has been happening for a while. So the European Medicines Agency has responded to charges of lack of transparency and concerns with proprietary frameworks applying to data collected during clinical trials by doing two things. The first one is COVID specific. So EMA has announced that data submitted by sponsors of COVID-19 vaccines and drugs, for that matter, will be published. So there's that requirement on sponsors of the successful vaccines that will come to market. They will have to publish their data. And this is part of a long-term policy. So it's not supposed to end with COVID, although it's right now suspended for non-COVID areas. But this is a long-term policy that includes much more than the release of just summaries. So you see the words here, clinical study reports. These are bits of information, quite extensive, that are submitted to the regulators as part of a new drug application or a biologics license application, as you would submit for a COVID-19 vaccine. And they are much more comprehensive than summary results. So if you're not familiar with these, to keep these in mind, so summary results are a couple pages, whereas clinical study reports range in the hundreds. And they describe the trial design, endpoints, methods, the data that have been collected, etc. So this is valuable information. that renders the process, the decision-making process on part of the regulator way more transparent, but also enables follow-on research. The beautiful thing about EMA having this policy is that the data submitted by Pfizer, Moderna, and other sponsors for COVID-19 vaccines is not going to differ very much between what they do in the US with FDA and what they do in other jurisdictions. So one jurisdiction pushing for greater disclosure of follow-on data might actually be something that has repercussions here, as we will be able to access that data, not from a perspective of justifying a particular FDA decision, but in terms of follow-on research, which is also important. I'm going to just end with a few considerations beyond COVID-19. So far, I've attempted to make the case that we are not yet very good at collecting data in the ways we should. We have a patchy infrastructure in terms of data collection, and we have, at least in the US, regulatory agencies, or a regulatory agency that further restricts through a proprietary interpretation of the law the ways in which we publicize the data used to grant things like authorizations for vaccines to come to market, and then full approval of these vaccines. But I've also tried to make the case that by conditioning this sharing of data, we might be impairing follow-on research. Obviously, I welcome, as somebody who thinks about ways in which we can improve biomedical research, I welcome the line of changes made by the EMA as opposed to the current FDA policy. And I hope that COVID-19 will inspire us to have more robust discussions here in the US about ways in which we could build trust in the data that we collect, in addition to fixing the structural problems with clinical trial data collection. But I also want to suggest that some of the things we're observing during the COVID-19 vaccine race might give us food for thought that extends beyond the context of data collection. The Pfizer and Moderna vaccines that we've been talking about are a completely new type of vaccine in terms of technology, mRNA vaccines, as opposed to the old school vaccines we used to have. They're produced in a context that's much more patent intensive, and I know the next panel will talk about this, than the older types of vaccine technology, for which there are fewer patents, further complicating the landscape of follow-on vaccine research. But now that we are about to have two very different types of vaccine technology, which is perhaps the most important moment in vaccinology that we can point to since the polio vaccine and the golden era of vaccine R&D, I think it's worth contemplating what's to come. And in COVID-19, in the context of the COVID-19 vaccine race, we've had machine learning used in the US and elsewhere to discover vaccine candidates. Currently, there are studies and clinical trials for cancer vaccines. These are occurring both in Germany and in the United States, in which we are going to soon, hopefully, turn vaccines into more instruments of personalized medicine by being able to target certain tumors and very quickly make a vaccine that caters to the specificities of certain types of solid tumors. So these are all things that are either being developed in connection with the COVID-19 vaccine race or preceded, but we should expect more types of vaccine technology to come to market within the next few years and decades. And the regulatory structure we have, the differentiation between EUA and full approval, and the ways we've keep, at least for the time being, data on these emerging technologies fenced in to some extent. This might be problematic down the road as the new forms of technology that have come to populate vaccine races are about to become much more diverse. So this is a good time to interrogate, I think, not just the ways we collect data and share it, but also the regulatory structure that we have in place to bring vaccines to market. And I'll stop there, and I really look forward to the Q&A and the upcoming panels.
Thank you very much, and an absolute tour de force by the four panelists. My huge thank you to each and every one of you for your outstanding presentations. We only really have a couple of minutes. Dr. Dean had a couple of questions. Professor Shah has a question too from Professor Brown. So Professor Shah, I'm going to turn to you first. And the question is, could you say more about the context-specific community engagement you had in mind for the ethical framework for your CHI studies?
Great question, Tennille. I think one of the big challenges with community engagement is that often there are lots of goals behind it, but the way you do it should be tailored to the reason for doing it. To me, the most important reason in the current pandemic is to address this issue of public trust. In prior studies that have involved community engagement, people have recognized that rumors can spread very quickly about challenge trials and can also influence how people feel about other kinds of vaccine trials. So Dorcas Kamuya in Kenya has done really incredible work creating not just a community advisory board, but an actual sort of deliberative body involving something like 200 community members where they share information about the study and there's a bi-directional relationship in terms of learning about research. And I think in the current pandemic, the other critical issue is engagement on social media platforms. When we submitted our article to Science, we included that, and the editor said, how can you possibly engage fruitfully on social media platforms? So it's an issue that's a little bit beyond my pay grade, but I think experts in community engagement can address that in an effective way.
Very good. Thank you, Professor Shah. You know, Professor Dean, maybe there were a couple of questions to you, and maybe if you can, in 1.5 minutes, give us your view in terms of the currently available interventions we have, ranging from vaccines to therapies. I think the audience would appreciate it.
Sure, and I have typed in some answers into the Q&A text. I think the most important therapeutic entity to keep in mind is the vaccine. I think that people may not understand that 95% effectiveness in a new vaccine is unbelievable effectiveness, and it is double what the effectiveness is of last year's influenza vaccine, which was only 45% effective. So in terms of evidence of efficacy, these two vaccines are unprecedented in terms of their initial efficacy. The antivirals and dexamethasone and antibody cocktails, they all have evidence to support efficacy in different settings. And I think that Lilly and Regeneron are the two leaders in the antibody cocktail mix, and both of them have filed EUAs for their respective drugs. Sometimes you'll read that they're not effective in combination with others, and that's because they're trying to put things together to get incremental improvement. I think I'll leave the therapies at that. Will, do you want me to make a brief comment about the other question?
Mike, in terms of public confidence, in terms of published data, yes. Would you spend 30 seconds on that? And then we need
the question was whether journals should speed up or relax their standards. And I think that they can speed up getting information out, but they should not be speeding up publishing data that are either very preliminary or flatly wrong. There was a Lancet paper that most people in our public remember as a fact, that hydroxychloroquine caused harm in a large population, and that paper was retracted because it was grossly flawed. We had a recent review of our hydroxychloroquine trial at the University of Utah by a community engagement committee, and they said, well, in view of the fact that the Lancet paper showed that this drug is harmful, why are you doing this trial? And this was the perception of people who are relatively expert 4 months after the paper had been retracted. So publishing an incorrect paper in this environment is harmful. I think the journals are just rushing to outscoop each other, which raises another ethical panel.
Thank you very, very much. Again, my huge thank you to each of the panelists. My thanks to you as the audience. I trust you found this session to be helpful to your understanding. Please stay healthy, be safe, wear your mask, social distance, and I'll put in my 2 cents worth for what we've seen from the vaccine data and their benefit risk profile. Please strongly consider an upcoming vaccine that I think the FDA will review carefully. So be safe, and thank you very, very much for being with us today. Bye-bye.